Structural basis of microtubule chromosome attachment during mitosis. Mitosis is a critical process in eukaryotic cell division, ensuring the faithful segregation of genetic material into two daughter cells. A key aspect of this process is the interaction between microtubules and chromosomes, which is mediated through the kinetochore, a proteinaceous structure that assembles on the centromere of each chromosome. The kinetochore acts as a dynamic interface that attaches to spindle microtubules, transmitting forces necessary for chromosome movement and orchestrating mitotic checkpoint signaling. This interaction is vital for maintaining genomic stability. Errors in microtubule chromosome attachment can lead to aneuploidy, a hallmark of several cancers and developmental disorders. The structural basis of microtubule chromosome attachment is an area of intense research, with recent advances in cryo-electron microscopy, super-resolution imaging, and molecular genetics providing deeper insights into the molecular architecture of the kinetochore and its interaction with dynamic microtubules. Understanding this intricate network of proteins, their conformational changes, and the regulatory mechanisms governing attachment fidelity is crucial for developing potential therapeutic interventions in diseases where mitotic errors contribute to pathology. This documentary delves into the structural basis of microtubule chromosome attachment, exploring its molecular composition, mechanistic function, error correction strategies, and implications in disease. The kinetochore is a multi-subunit protein complex comprising over 100 different proteins, organized into distinct layers that establish a robust yet adaptable connection with microtubules. Structurally, the kinetochore can be divided into the inner kinetochore, which is anchored to centromeric chromatin, and the outer kinetochore, which extends into the cytoplasm to interact with spindle microtubules. The inner kinetochore is primarily composed of the centromeric nucleosome, which contains a specialized histone H3 variant called CENP-A. This epigenetic mark is crucial for recruiting other centromere-specific proteins including CNPC and CNPT, which serve as scaffolds for outer kinetochore assembly. CENP-A containing nucleosomes provide the structural foundation for kinetochore formation, ensuring that the centromere remains a stable site of attachment across cell divisions. The outer kinetochore consists of the KMN network, a core microtubule binding module made up of the KNL1 complex, the Mis12 complex, and the NDC80 complex. The NDC80 complex is a critical component, directly binding to microtubules through a calpanin homology domain in the NDC80 subunit. High-resolution structural studies have shown that the NDC80 complex extends as a rod-like structure with flexible hinge regions, allowing it to form multiple contact points with microtubules. This interaction is further stabilized by the presence of microtubule-associated proteins such as the SCA complex, which enhances the persistence of kinetochore microtubule attachments. The dynamic nature of microtubule attachment is facilitated by the ability of kinetochores to undergo conformational changes. Studies using cryo-electron tomography have revealed that kinetochores can adopt a curved conformation when attached to depolymerizing microtubules, suggesting that they can harness microtubule depolymerization as a source of mechanical force for chromosome movement. This structural flexibility is essential for maintaining attachment under varying mechanical tensions during mitosis. Microtubule capture by kinetochores is an essential step in establishing proper chromosome alignment at the metaphase plate. Initially, kinetochores encounter microtubules through lateral interactions, in which the kinetochore glides along the microtubule lattice before transitioning to a more stable end-on attachment. This lateral to end-on conversion is a critical step that ensures robust connections capable of withstanding mitotic forces. The conversion to end-on attachment is mediated by multiple microtubule-associated proteins, including the SCA complex and the DAM1 complex in yeast. The SCA complex acts as a molecular clamp that enhances kinetochore-microtubule interactions by binding along the microtubule lattice, stabilizing the connection even as microtubules undergo polymerization and depolymerization dynamics. In contrast, the DAM1 complex forms a ring-like structure around microtubules, coupling kinetochore movement to microtubule dynamics. Once end-on attachment is established, kinetochores must resist premature detachment while still allowing microtubule dynamics to drive chromosome movement. This is achieved through a balance of stabilizing and destabilizing forces. The NDC-80 complex, in conjunction with the SCA complex, provides a firm grip on microtubules, while regulatory proteins such as Aurora B kinase introduce reversible phosphorylation events that modulate attachment stability. This dynamic regulation ensures that kinetochores remain attached to microtubules while also allowing erroneous attachments to be corrected. Accurate chromosome segregation depends on the ability of kinetochores to detect and correct improper microtubule attachments. Errors can arise from monotelic, meaning a single kinetochore attachment syntelic, where both sister kinetochores are attached to the same pole, or merotelic, which involves one kinetochore attached to microtubules from both poles. If these errors are not corrected, they can lead to chromosome missegregation and aneuploidy. Aurora B kinase plays a central role in error correction by phosphorylating key kinetochore components to destabilize incorrect attachments. The spatial positioning of Aurora B within the inner centromere allows it to act as a tension sensor. In cases of incorrect attachment, low tension results in high Aurora B activity, leading to phosphorylation of the NDC-80 complex and subsequent detachment of microtubules. Correct bio-oriented attachments, where sister kinetochores are pulled towards opposite poles, generate high tension that moves kinetochores away from Aurora B, reducing phosphorylation and stabilizing the attachment. Additional regulatory mechanisms, including the Spindle Assembly Checkpoint, or SAC, monitor kinetochore microtubule attachment status. Unattached or improperly attached kinetochores recruit checkpoint proteins such as MAD1, MAD2, BUB1, and BUB-R1, which inhibit the anaphase-promoting complex, or APCC, to delay mitotic progression until all chromosomes are correctly aligned. This checkpoint ensures that cells do not prematurely enter anaphase with erroneous attachments. Defects in kinetochore microtubule attachment mechanisms are implicated in numerous diseases, particularly cancer. Aneuploidy, resulting from chromosome missegregation, is a common feature of tumors and contributes to genomic instability. Several oncogenic pathways are known to disrupt the fidelity of microtubule attachments, leading to increased rates of chromosome missegregation. One of the key proteins often deregulated in cancer is Aurora B kinase. Overexpression of Aurora B leads to excessive phosphorylation of kinetochore substrates, increasing the likelihood of unstable attachments and chromosome missegregation. Conversely, loss of Aurora B function results in failure to correct erroneous attachments, leading to persistent missegregation errors. This has made Aurora B an attractive target for cancer therapy, with small molecule inhibitors such as bericitinib and alisertib being developed to selectively target its kinase activity. Microtubule-targeting drugs, including taxanes, for example paclitaxel, and vinca alkaloids, for example vinblastine, also exploit the mitotic machinery for therapeutic intervention. These drugs disrupt microtubule dynamics, preventing proper kinetochore attachments and leading to mitotic arrest and apoptosis in rapidly dividing cancer cells. While effective, resistance mechanisms such as upregulation of microtubule-associated proteins pose challenges for long-term therapeutic success. Emerging strategies aim to target specific kinetochore microtubule interface components with greater precision. Small molecule inhibitors of the NDC-80 complex are being explored as potential antimitotic agents that disrupt kinetochore microtubule attachments without affecting other microtubule functions. Additionally, checkpoint inhibitors targeting SAC proteins are being investigated as a means to selectively kill cancer cells with defective mitotic checkpoints. The structural basis of microtubule chromosome attachment during mitosis is a finely tuned process involving a network of protein interactions that ensure accurate chromosome segregation. The kinetochore acts as a dynamic interface that captures, stabilizes, and regulates microtubule attachments while also integrating checkpoint signaling to prevent errors. The complexity of these mechanisms underscores their importance in maintaining genomic stability, and their disruption is a key driver of tumorigenesis. Advances in structural biology and molecular genetics continue to shed light on this intricate process, paving the way for novel therapeutic strategies targeting mitotic errors in disease.